Good evening, folks. For those of you who are new to Wolfson, welcome. For those of you who are here regularly, it's nice to see you again. And as most of you know, this year is our 50th anniversary, and one of the ways we are celebrating this is by commissioning a series of monthly lectures by distinguished members of the college, and in which we ask them to look back on what has happened in their specialist fields during the half-century that Wolfson has been in existence. This month's lecture has been given by Professor James Wood, who is a Fellow of the College and Head of the University's Department of Veterinary Medicine. He joined the department in Cambridge in 2005, initially as Director of the Cambridge Infectious Diseases Consortium, and in 2009 he was appointed the ALBORADA Professor of Equine and Farm Animal Science. He's a veterinary epidemiologist by background. He studies the dynamics of emerging infectious diseases, including viral infections in fruit bats in West Africa, focused on Ghana, mammalian influenza, rabies, and bovine tuberculosis. This is laugh-a-minute stuff. He has an MSc in epidemiology from the London School of Hygiene and Tropical Medicine, and a PhD from the Open University, which he completed when he was head of epidemiology at the Animal Health Trust. He's also a graduate of the Royal Veterinary College, and to top it all, did a BSc in physiology at UCL in the 1980s. He's here this evening to lecture on an intriguing topic: bats and rats and emerging infectious diseases. Ladies and gentlemen, please welcome James Wood.
Thank you very much, John, for the generous introduction, and particularly for the introduction, the invitation to talk this evening. I'm going to talk about the title is Seed Bats and Rats and Emerging Infectious Diseases, and try and give a perspective on how some diseases have emerged from such species as bats and rats over the last 50 or so years, and to try and give a perspective on how we might go about studying these things, these occurrences, which can be somewhat challenging and under rather challenging circumstances, because many emerging infectious diseases, there's a tendency for them to emerge in the tropics where things are further from stable infrastructures, which can make their study harder than just being able to simply study things within a laboratory in Cambridge or other places around the country or the world where people work on these things. I'm going to give a personal perspective on this, but I want to start by acknowledging the huge number of collaborators that I currently have working with me in our work in Ghana that you just mentioned in the introduction. In particular, Richard Suuri, that we worked with for the 10 years that I've been working in this area, who's a wildlife vet in Ghana, and an ex-Pro-Vice Chancellor of the University of Ghana, who's a distinguished zoologist, Ya Ntumo Bedu, who has been a great collaborator for us in the University of Ghana. And many other social scientists and natural scientist collaborators that have contributed to much of the work we're talking about. And of course, we're also very reliant on our funders, as well as the specific funders, some of whom that I describe here in this slide, from the European Union and from UK government research funding. I'm also very lucky to be the recipient of a very generous support from the ALBORADA Trust. So thinking about looking back, many emerging infectious diseases, diseases have emerged over the last 50 or 60 years, and these two graphs, working from the 1940s through to the previous decade, have really demonstrated that the rate at which diseases have emerged has increased over this time. I think it's very important that we're careful to consider the difference between true emergence and the rate at which diseases have genuinely newly developed, and our ability to detect them. And I think that graphs like this really are. It's important to note that they're corrected for the rate that, at which we're getting, the fact that we're getting much better at detecting them. And so some of this increase in emergence relates to an increase in diagnosis as much as an increased burden of disease caused by these things. But some very important features of disease emergence, and I'm talking about infectious disease emergence, because obviously many diseases such as diabetes have emerged over a similar sort of time period, but that relates to lifestyle, as a previous lecturer in this series has talked about. But many of the new diseases, new infectious diseases that we have to deal with in the human population, as well as in the domestic animal population, have so-called zoonotic origin. So many of our diseases, far more than half of them, come from animals. And a really important feature of this also is that many of the infectious diseases that have emerged over this time from animals have come from wildlife, not just from domestic animals. And it's obvious that there are far more wildlife species than there are domestic animal species, but it's also true that there is a far bigger gap in the way that we live. We live, at least parts of the human population live very closely with domestic animals, but as a generalization we live far further apart from wildlife. Apart from intriguingly, bats and rats. There's a story that you're never more than 10 feet away from a rat. I think that's in Oxford, not in Cambridge, but it's certainly true in London. And actually, in many parts of the world, we live very closely with bats who will live in the roofs of our houses. I'm going to be talking more about fruit bats that tend to live further apart from us, often in rather large roosts or colonial roosts. But it's not a... But these around these roosts, these can be in very close proximity to the human population. As I think you'll see from some of the pictures I show, given the numbers and weight of these bats, that sometimes in the developed world might seem rather surprising to us. I think a very important feature, though, over the last, on this slide obviously goes back for more than 50 years, but over this time period, the proportion of those diseases that are emerging from wildlife has been increasing, and I think it's clear that relates to the way that we live differently in the world. We're forever encroaching on what were historically more remote rural areas, and when we do encroach on them, often we travel back very quickly to centres of more developed centres, where our ability to diagnose these infections will be far greater than being stuck out in the middle of a rainforest or in a small, rather remote village. But why bats? So one of the reasons that we might consider bats, sorry, I've jumped a slide, is the fact that they are the hosts where viruses persist from a number of virus families which are known to be particularly pathogenic. And I'm going to refer to some of the textbook dogma that that exists in relation to these diseases, and I think there's a really strong dogma surrounding rabies virus, and something that most first-year medical and veterinary students and the general population are aware of if they know anything about rabies, and that is, if you get it, then you always die. And I think that's something which is true across many species, including ourselves, and there's probably a nearly always that should be added there, but actually that's not true for bats, and it's quite an interesting feature of many of these diseases. Now, the filovirus family, including Marburg and Ebola virus, are thought to kill pretty much everything that they infect, and until the current awful West African epidemic of human Ebola, where now survivors from that epidemic are recognised as being not uncommon. It was thought that the vast majority of people that were infected with Ebola virus also died. Two viruses I'm going to talk a little bit more about, because they've been the subject of our particular study in West Africa, the Hendra and Nipevirus, that form the Henipavirus family, or at least the two main representatives of the Henipavirus family. These are viruses that have emerged in the last 20 or so years, and I'm going to talk specifically about how they've emerged. But in many cases, these have also been associated with a very high case fatality rate in humans that have been unlucky enough to be infected with them. Of the coronavirus family, the SARS coronavirus, which emerged and caused devastation around the world, and particularly in Taiwan, Hong Kong, and southern China, but also because of spread through very effective spread through air traffic. Also parts of Canada and Europe and other parts of North America as well. SARS coronavirus was also associated with a very high case fatality rate of around 40 or 50% of those people who were infected, certainly in some parts of the world, who then subsequently died from it. So we fear bat viruses because so many of them have such a high mortality rate if they infect humans. But are bats really so much worse than other mammalian groups? And I think this is for us has been a very, very important question. It's clear that some bat viruses are extraordinarily nasty or pathogenic. But shouldn't we expect that for a group of species that comprise 20% of all mammals? Now that's not all mammals in relation to numbers of individuals that live, but certainly all mammalian species are bats. 20% of them are bats, which are, again, for those of us who live in the northern hemisphere, actually that's rather surprising because we hardly ever see them. And of course the reason that we hardly ever see them is they tend only to come out at night. So when we do see them, you just see a kind of fluttery wing, and you assume it's just the bat species that lives next door. But actually, there are quite a wide variety of them. And the other issue that we might think about with bats is whether there are in fact any worse than rodents, which have been long recognised as a source of infectious diseases, be it plague going back many centuries, although there's still foci of plague that exist in wildlife to this day. But Lassa fever, which is a very extraordinarily nasty hemorrhagic fever that persists in West Africa, particularly Nigeria and Sierra Leone, finds its home in multi-mammate rats, and hantaviruses are found in Europe and North America and South America in a variety of different rodent species. And so how do we think about comparing bats and rats and whether one is really worse than the other? And what we did with what our postdoc Angie Lewis did, who was based at the University of Colorado with collaborators there, conducted a systematic review of all viruses that were reported in the medical literature that were found in bats and rodents. And when looking systematically, then you can start counting them. And in fact, what's very interesting is that per bat species, you get more viruses in more zoonotic viruses, those viruses that can spread from bats to humans per species than rodents. But because there are so many more different rodent species than there are bats, actually there are more zoonoses in rodents found than there are in bats. So this idea that bats are the source of all evil, I think is probably a little bit unfair. And the rats and mice found in the many different species found in the rodent group are actually the more common hosts of more viruses. But what's interesting, I think, and probably very, very important in terms of understanding persistence and transmission, is that you get more zoonotic viruses hosted by species who are overlapping geographically with lots of other bat species, and that's I think a very important feature to think about, because it's not enough just to have a virus that can transmit. It's also got to be able to transmit and pass between different species, and often it will pass between a number of different species before it infects us, and so those bat hosts that are overlapping with many other bat hosts, obviously in very biodiverse regions, and that often is in the tropics, but not invariably, will often be hosting more zoonotic viruses than those that are found in a lower population density or lower species densities. And so I think that this raises some very important questions, which may actually be particularly interesting for virus families such as Ebola. That if you're going to get viruses that transmit to humans, in many cases they will already be transmitting between different wildlife species first. But what are the diversity of viruses that exist in different groups of species? Now, I'm going to talk a little bit about the lyssaviruses and then the Nipeviruses. And the lyssaviruses are the family of viruses that include rabies as the prototype virus. But in fact, there's a massive diversity of viruses, particularly infecting different bat species around the world and many different parts of the world. That are found, the great diversity found in bats around the world, and some, and nearly all of the different groups represented by different notations here are found in bats. But by far the greatest burden of disease is caused by rabies virus. And although some of these rabies viruses do infect bats, particularly in North America, by far the greatest burden of disease in humans is caused by not bat rabies viruses, but simple rabies virus in domestic dogs. This is where I think it's very important to distinguish numbers of viruses and potential threat from the burden of disease that you see in human beings. And this is a very important distinction in public health terms. So the rabies viruses that persist and infect in domestic dogs are very well adapted to spread between dogs. And unfortunately, if a dog bites a human rather than another dog when rabid, then that will result, if that human being is untreated or unvaccinated, will result in that human developing rabies and usually dying from it. We've been focusing on African lyssaviruses, in particular Lagos bat virus, which has a number of different subtypes within it. This is a virus that has been shown to spread occasionally to other domestic animal species. Cats and dogs have been affected and so on. But it's not one that's widely recognised as causing a major burden of disease at this time in Africa. But you can see that there's a massive diversity of different viruses here. In contrast, the Nipeviruses, which have only been identified since 1994, so only just over 20 years, these first emerged as a new virus family, the first virus obviously started as a family of its own in terms of our recognition, in 1994, where there was an outbreak of respiratory disease in a racing yard just outside Brisbane, in a small suburb called Hendra, which I think is named after an even smaller village in North Cornwall on the coast. This outbreak killed 13 horses and the trainer. 2 other people, the vet and one of the stable hands, also became infected. The vet became very ill but recovered. And retrospective investigations then found that this was preceded by 2 horse deaths on a stud farm in which the farmer also died. The farmer had helped with the post-mortem examination of those 2 horses. It's the first cousin of Hendra virus, Nipevirus, first emerged in 1999, some 5 years later. And I'll talk a little bit about both of these emergencies. But this emerged as a disease of pigs and pig farmers and slaughtermen in Malaysia and Singapore in 1999, and was associated with the death of more than 100 humans and the slaughter of of more than 1 million pigs, which were slaughtered to try and bring the outbreak under control. So how do these diseases first appear to us? And I think Hendra is an interesting example, because there was an outbreak of fatal disease in a racing stable in Brisbane. The trainer was said to have some interesting connections with different betting syndicates, and it was thought that he might have upset 1 or 2 of them, and because of that perhaps unfounded suspicion, poisoning was suspected, and the horses were examined post-mortem in the stable yards. Apparently they were just cut up in the middle of the yard and samples collected for toxicological investigation, but a few were also kept for a more standard examination. And after the first few horses had died, then suddenly the trainer became ill as well, which put a very different complexion on this. And you can see a headline here from the Independent in September '94. And when a few days later, when the trainer died, then people started to look much more carefully and broadly to see what might have been the cause of the death of the trainer and the horses. And then a few days later, you can see that was the 7th of October, so some 10 days after the trainer first died, it became clear that from virological investigations that Vic Rails had been killed by the same virus that it was associated with the death of those horses. The other horses in the yard, as commonly happens even in this modern world, were killed by human hand in order to stop further spread of this infection around either to other humans but also to other horses. So then the big question then came, where did Hendra originate? And so this was in Brisbane in Queensland, which is the northeastern part of Australia. There were huge investigations of every animal species, that wildlife and domestic animal species in Queensland over the next year or 18 months. And all other so-called terrestrial animals, you might say, were found to be negative serologically for Hendra virus. So there was no evidence of widespread infection around animals in both in domesticated animals but also in wild animal species. But someone had the bright idea of looking at a few species of flying fox, the big fruit bats that live in across the coastal parts of Australia, and found that the infection was really very common in all 4 species of fruit bat. It was rather harder to find a virus itself rather than just the antibodies, evidence of prior infection and immune response to it in the bats. And it was interesting that it wasn't found in the insectivorous bats that were in the same regions, so-called sympatric, that those living in the same area as the infected fruit bats. And this provided very strong evidence that the fruit bats were the reservoir, were the source of the virus for the horses and for the trainer. And this rather unpleasant disease then disappeared for 5 years. And I think everyone rather heaved a sigh of relief and thought, well, that was a one-off, and the horses got it from the bats. Not quite sure how they did, but never mind. It's obviously not going to happen again. And then 5 years later, there was one horse that was diagnosed with this really rather unpleasant disease, which is associated with an overwhelming pneumonia in most cases, but also with an encephalitis and a few subsequent outbreaks, particularly those that have occurred subsequent to 2007, 2008. And most of these spillover events were associated just with the death of a horse, but the lucky recovery of a vet in 2004 who just had a relatively mild respiratory illness. And then there was an outbreak in a few horses that were hospitalised in a Brisbane veterinary hospital, and perhaps because of biosecurity and late diagnosis, the main vet in that practice caught the infection from them. There was severe illness in the veterinary nurse, who 2 years later was learning how to speak again, the extent of the encephalitis. And a second veterinarian was hospitalised, but recovered with far less sequelae than the nurse suffered from. So it was this outbreak that really has changed to a great extent the whole nature of the horse industry in Queensland. And horses used to be everywhere, and people had a very relaxed view of riding, and it's what everyone did. And suddenly now in Queensland, the horse is framed as being far more of a worrying animal to look after. And it's the fear of these very unpleasant zoonoses that can have sometimes, in societal terms, as big an impact as the actual disease itself. And it's a really important feature. And you can see here that there were just a few cases every year after into the 2000s. But then, for reasons that are not fully understood, but probably to do with the massive flooding and climate extremes that occurred in 2011, there were 17 spillover events in 2011. Luckily, the Australian veterinary profession, the equine veterinary profession, has got a little bit smarter about dealing with these things now, and biosecurity and personal protection for horses that have any sort of suspicious signs in this region are now dealt with very, very differently and managed very, very differently. The veterinarian who's the head of the hospital that first examined the horse back in 2007, as he narrated before he died, that he knew he had personal protective equipment in the car, but after he'd taken a look at the horse, he didn't want the owners to think that he was a bit of a softie to go back and put it on. And actually that story has left a really big mark on the way that people have approached this, because it's No longer is it regarded as being a softie to take proper care of yourself and your staff, which is a pretty good change. So there was an explosion of events in 2011 which thankfully didn't result in either much or any horse-to-horse transmission, but any transmission to humans. But certainly over the last 20 years, there have been the deaths of these 83 horses and 4 people over this time, and that's not a huge number of horses. It's not a huge number of people, but it's had a profound impact on society. And you can see that these are all very coastal outbreaks. There have been a few cases, most of which were in 2011 in northern New South Wales, but they're all in coastal areas. And perhaps not surprisingly, the greatest risk for spillover is where there's an overlap between high areas of horse density and bat density and flying fox density, as the Australians refer to their large fruit bats. The Nipevirus outbreak in 1999 was quite different because it didn't involve horses at all, but there were, as a result of an outbreak of respiratory disease on a major pig farm in the centre of Malaysia. This was a really big breeding pig farm that produced piglets that were sent all over the rest of the country for fattening. It was much easier to fatten pigs than it is to actually breed them in a high rate. So, because this is a central farm to the pig industry in Malaysia, not only did it send its pigs out around the country, it sent pigs with Nipevirus in them all over the country, which is why so many people became infected as well. But as a result of this outbreak, there were more than 1 million pig culls, 800 pig farms were demolished, and thousands of jobs lost acutely. Now, there's been a bit of a regrowth of the Malaysian pig industry there, but there was a massive economic burden as a result of that animal disease, and obviously widespread human disease. And this was such an intensive farm that once the infection got into pigs, hundreds in a pen, it can spread very, very easily between them. And the reason you can see the density of buildings here and reflected by the density of buildings around this whole site, surrounded by fruit orchards. The fruit orchards actually encroached right over the top of the pig pens, and where these arrows in this slightly blurry picture, sorry, I haven't moved on, where the arrows, the fruit trees encroaching over the top of the pig pens, where these arrows indicate actually were just roosts of fruit bats, which were right over the top of the pig pens, which were underneath fruit trees, in many cases mango trees. And the thing about a fruit bat is that it will pick the ripest fruit, and in many cases then it's eating, and its salivation all over the fruit will result in the fruit dropping off. And there's nothing a pig likes more than a really ripe mango. And so as a result of fruit falling, of half-eaten fruit falling, this is almost certainly the route that pigs got infected. And if it wasn't by this route, it's the fact that one of the best sources of virus, if you're trying to sample from bat populations, is their urine. It's very difficult to collect urine from underneath a very dense bat colony. You need a plastic sheet and a syringe. And if you go there early in the morning before when the bats fly back from feeding, typically before they go to sleep, they have a pee, and you can then collect that and try to avoid being watered on yourself in the process. As well as this really big outbreak, which is a one-off in Malaysia and Singapore, this virus, it turns out, has been emerging constantly in Southeast Asia, and there are our best reports for the first decade of this century, where there were lots of outbreaks where there was a transmission from bats to humans in many different villages across the belt, where there is a high density of fruit bat populations. And the real difference feature of these rural Bangladeshi outbreaks is that there's quite a lot of direct human-to-human transmission as well, but that reflects the way that people traditionally look after their sick family members, which traditionally is done by very, very close nursing, and often sharing a bed with, trying to keep them warm and so on when they're feverish. And so in any case, despite this, there's been no long chains of transmission that have gone for weeks or months, but a lot of transmission which eventually has been self-limiting, often unfortunately because of the fact that around half of the people infected in Bangladesh with this virus have died of it. So I hope that those two stories give a feeling of how fatal emerging infectious diseases can emerge in the developed world, but also in the less developed world. And I think it's really important to think about the impact. It's not just mortality, but it's also the reaction to the event as well. And there are often, frequently, and this is really evident with the West African Ebola crisis, massive societal impacts of emerging infectious disease, often relating to fear and societal change, as well as the actual mortality itself. So I think that provides a justification for why we should work on emerging infectious diseases. But then we do have to think about what we can do about addressing the challenges of emerging infectious diseases. And then also we've got to think about what we can get funded, because and answering some important biological questions, as well as trying to reduce the burden of these diseases. So one of the reasons that bats and rabies-like viruses are really interesting is that so many bats don't die when they get infected. And so bats haven't read the textbooks and don't do what they're meant to do, but it's really interesting to try and understand why that's the case. And that's some strong evidence that's come from the work that we've done, but also other groups have done, looking at these lysoviruses, particularly Lagos bat virus and fruit bats in sub-Saharan Africa. And what you find is in older bats, and you can age them by looking at the number of rings that they have in their teeth, because they lay down a ring for every year, is that certainly in our population, more than 3/4 of the older bats will have been infected with this rabies-like virus, and the prevalence of prior infection is much smaller in younger bats. So this is a prevalence that increases with age, and it's clear also that for bat species that are infected with Ebola virus in sub-Saharan Africa, that it's quite common to find bats that have recovered from it. And although the recovery is less well characterised than it's been possible for Lagos bat virus, but it's clear that from a very small study that we published, that bats can survive quite a long period of time, despite the fact that they have been previously infected with Ebola. So how do they do that? Why don't they die? And certainly at the individual level, there are almost certainly immunological mechanisms that underlie that. Maybe they've got a particularly well-adapted innate immune response that enables them to cope with viruses that are very pathogenic in other species. And there are lots of groups working on that. That's not been a particular focus of our group's work, but although we provided a lot of samples for groups doing that sort of work, we've been more focusing on trying to understand the persistence at the population level for a virus that is so often pathogenic and fatal. It kills animals quite quickly when they do get ill. How does it transmit at the population level? And that's a question that we've been trying to answer. And as well as that, we've been trying to understand how these viruses may spill over from bats into humans or domestic animals. So the challenges we're addressing is persistence of pathogenic viruses in bat populations. And understanding some of that relates to the need to understand viral diversity, because although traditionally we thought about a virus as being, well, that's a virus and that's its sequence, what you find in the real world is something very different to that, particularly with these typically RNA viruses, is that they're actually, because they're rather poor at replicating themselves, their replication machinery makes lots of mistakes. And so what you get then is a very noisy, messy sea of viruses that are all very closely related, but actually also different in many different genetic ways. And we've been trying to understand, and other groups are trying to work on the diversity you see in viral populations in reservoir hosts, to see if that underlies some of the ability of those hosts to host these highly pathogenic viruses. And spillover is something that we've been looking at directly ourselves, trying to understand whether human populations in close contact with large roosts of bats have been infected. One of the greatest links between humans and bats, as well as the rain that you get every morning underneath the bat colony, is bushmeat, because large fruit bats are eaten quite commonly in sub-Saharan Africa. Now, we chose to work on the straw-coloured fruit bat, Eidolon helvum, This is a large colonial sub-Saharan fruit bat species. Individuals have been tracked migrating over a few weeks, 6 weeks or so, where they travel for more than 2,000 kilometres. So these can travel very long distances. And the reason that we've picked them is it's a common bat. It's clearly able to live with rabies viruses. It's infected with African Nipeviruses. So straight away it makes it an interesting host because it's got more than two quite different virus families that are commonly infected. It's commonly eaten, particularly in West Africa, not so much in East Africa, although in Pemba, it's one of the islands of Tanzania, it's widely consumed as well. It's interesting because it's migratory, which creates great opportunities for continental spread, and lives in huge populations of up to 10 million in one case. I'm going to show you a bit of film of one of those. And there's little multidisciplinary study of these populations. So this is what the sky looks like when bats start flying at dusk. And just that single quite high quality picture doesn't really capture what happens at dusk when the light gets dimmer. And I'm going to show you a rather poor footage here, but listen to what you're surrounded by when these bats take off. They're quite noisy. So this is a population in Kasanka Park in Zambia where for 2 months of the year, every year, there are 10 million of these that converge on Kasanka Park and then fly away again. But every evening, for around an hour or so, these bats take off from the trees that they're living in and devastating because 10 million bats, each of which average 250 to 300 grams, is several hundred tons of biomass of meat. So you can imagine that's quite a lot for the trees there to support. These are interesting species to live with. And so we've been looking, and I'm going to just run through rather quickly a series of different types of studies in a multidisciplinary program that we've looked at. We've been looking at studies of naturally living bats. We've kept a small experimental colony in Ghana in a cage that we built 5 years or so ago. We've been looking at bushmeat, its consumption, the commodity chains, and also human studies, both thinking about infection in people that live close with bats, but also working very closely with social scientists, social scientists working as part of our teams, trying to understand how people live with bats, what are their perceptions of them, and actually what happens in relation to health-seeking behaviours in the parts of the world where people live very closely with these large roosts of bats. So think about the population dynamics, and bats are quite different. Living these numbers are rather difficult to study because they all look the same. Not to them, but to us, they all look the same. And because there are so many of them, if you try and capture the bats in the same colony repeatedly using mark recapture approaches, which are standard in wildlife ecology, you never catch the same bat twice because out of 1 million bats, you're so unlikely to recapture the same individual. So it makes it really challenging. So the way that we did a mark recapture study was to put radio collars on 100 bats that had an individual ping that a receiver could pick up, and they had batteries that lasted for 1.5 to 2 years. And then we walked around the colonies, our collaborators walked around the colonies where we were studying them to see what proportion of them were still there and so on. And what we found, and so you walk around waving a large television aerial under them, which is a strange thing to do in the middle of an African city, but what we found was we put them on in the colony in Accra and then studied them, and what we found was that the numbers of bats fell very rapidly after we put these radio collars on, and by the end of March, April, they'd nearly all gone. And they'd nearly all gone because they migrated, and we knew they migrated, which is why we'd rushed to get the collars on beforehand. And what was very interesting was next year, not very many of them came back. And only around a fifth of them came back. And then those migrated away again 3 or 4 months later, at the end of the dry season. And What's interesting is that those numbers, because we use this method, which is quite precise, to estimate the population size, or the relative population size, which we combined with straightforward roost counts, going around all of the trees in the roost and trying to estimate, based on extrapolation, the number of bats in those roosts. What we then find is that the numbers that came back, they started at around 1 million, only 250,000 or so came back at the next dry season. Then there were 2 or 3 years like that, and then another year, around 1 million or so came back. And so it wasn't that there had been an 80% mortality. This reflected the fact that these populations of bats shift all over the subcontinent. We've looked at the age structure of the population. We find the average age of the bats in this roost is around 7 years, but some of them may live up to 14 or 15 years. And we've looked at them all across the continental range of these bats. And as I said, they all look the same within a roost, and they all look the same between the roosts. So these are pictures taken from Kumasi in Ghana and Kasanka Park that I just showed you that video from in Zambia. And in order to understand how they relate to each other, because they're a little bit too small to put on the heavy batteries that we need to have satellite transmission going back with GPS loggers on them, we've looked genetically at the connectedness between Eidolon Helvum roots, and have used both mitochondrial markers, the markers that we all inherit and bats inherit from their female parent. And what we found was that there is of the continental bats, we found that in this haplotype network, all of the continental bats are absolutely muddled up. What's absolutely fascinating, and if I just go back to the map and show you, the colonies that we sampled in 3 remote islands off in the Gulf of Guinea, off the coast of West Africa, these remote island populations in Annobon and the 2 close together islands of Principe and Sao Tome were genetically quite distinct. And when we take what's referred to as a microsatellite marker, which is looking across all of the genes of the bats rather than just that subset of genes that are inherited from your mother, what we find is that there were 3 populations. So there were the continental bats, there was the bats from Principe and Sao Tome, and the quite distinct family that come from Annobon. And there was a very clear signature that demonstrated that was that we had these 3 very distinct populations. Now, the fact that the islands were different wasn't that surprising, but what was really surprising to us is that across the whole continental range, you were as likely to find a related bat in the same tree as you were from one that was from Blantyre in Malawi all the way up to Accra in West Africa. And there was no hint of genetic, what we call separation by distance across the whole range. This makes this one of the genetically best mixed species that's ever been studied, bar, I think, 1 bird and 1 mammalian, 1 sea mammal species, which are known for their widespread genetic diversity over huge geographic ranges. So we looked also at how infections are spread at the population level. And we started off by considering, just very simply, does the seroprevalence range? Now, if we look at the proportion of bats that have been previously infected in these different populations, what you find is there's quite a lot of variation. In many cases, it reflects the fact that when you sample from one colony, the age is very different, of bats is very different in that colony compared to some of the others. But all of the bat colonies that have been looked at for both Henipaviruses and Lagos bat virus have been widely infected. And when we consider these analyses, when we look at the rate at which these animals get infected over their lifespan, what you find is that after the apparent loss of maternal immunity, which you find in very young bats, the older they are, the more likely they are to be infected. And that's true for both Lagos bat virus and also for Henipaviruses. And with some interesting but subtle differences that you might expect between those 2 different families. But are the viruses exactly the same in the different parts of the continent where we've looked at them? Now this virus family tree, which you can see is divided into two parts, shows some of the virus diversity, the paramyxovirus diversity that we found across the subcontinent, across the continental range. And I'm going to break this tree down into the two component parts so you can see it. But this contains all of the paramyxoviruses, not just the smaller Nipevirus family that exists within the paramyxovirus family. And what you find is that the viruses, or the bit of genetic sequence of the virus that we were able to capture in these urine samples was the same, or there was a massive merging of it, whether it was a virus that's very close to mumps virus, which was very similar in Tanzania or in Ghana, or the slightly more diverse viruses that were found in Ghanaian bats, or in the shaded area, Ugandan bats. And really there's no geographic difference at all in that part of the family tree. And similarly, in this other part of the family tree, which again is slightly closer, but still quite some way from the Nipeviruses and the Hendra viruses. We find that Tanzanian and Ghanaian bats, so East African and West African viruses, were really closely related to each other, with absolutely no sense of geographic separation, which is entirely consistent with the way that these bats migrate. So that told us a little bit about some of these viruses, starting to give us hints about how the viruses may persist in these bat families. We've also, working very closely with the Wellcome Trust Sanger Institute just down the road from here in West Cambridge, we've also used genomic technology to try and discover many different virus families. And we've taken samples from the lung, from the throat and from the urine of 60 or 70 bats that were sampled from Ghana, and used cutting-edge molecular and genomic techniques to try and identify virus families. And one of the greatest challenges with the sequencing blind from samples collected from any species is removing all of the host genome, because the vast majority of the genetic material that you sequence actually just reflects the fact that you're sequencing bits of the bat host. But working through massive bioinformatic pipelines, which are kind of almost harden the technology of the sequencing, we did indeed find, perhaps unsurprising, but a wide variety of different virus families within this one species of bats. So we found two different herpes viruses, evidence of both a beta and a so-called gamma herpes virus. We found the papillomaviruses, the viruses that are associated with warts and some cancers. We found quite a wide variety of different papillomaviruses, particularly in some of the throat samples from the straw-coloured fruit bat. We found and actually managed to isolate an adenovirus, which fitted into the middle of the adenovirus family. Perhaps quite surprisingly, certainly it was surprising to us at the time, we found a very strong signature of a pox virus. It's very closely related to Molluscum contagiosum that can be a real pain for young children, as many parents know. It's quite a slow to a virus that causes quite a chronic infection in children, and certainly my experience of our young children was that once they got it, they took a long time to get over it. Well, this is thought to be a unique human pathogen, but in fact, the virus that we characterised was the closest relative to this virus, and that's in an African fruit bat. So then, these are the viruses, but what about spillover and detection? So I mentioned bushmeat earlier in the talk, and I'm just going to talk to you very briefly about some of the bushmeat studies that we've done, but this picture really encapsulates some of the interesting features of bushmeat. So this is a chap who's slingshotted a few bats out of the large roost in the capital city of Accra. He's selling them by the side of the road. You can see that he's not. He's not wealthy, and that's a common feature of those that hunt bushmeat. Often it's the poor, often it's the rural poor, but it's often a practice, a livelihood associated with poverty. You can see here that there's one of these bats that's not actually dead, that's still moving around on the pavement. These bats will be quite aggressive, obviously, at this stage once they've been pulled out of a tree, and there's a distinct risk of these bushmeat hunters being bitten by bats, and frequently they report being bitten by bats. And perhaps an unusual feature of this particular picture, which I'm going to tell you about, which you can't see, is that I've never seen these being sold by the side of the road. It's really uncommon to see these bats being sold on the side of the road because they're so popular. They've gone as soon as they land there. But because it's such a popular form of bushmeat in Ghana, at least it was until 2 years ago. That's a feature of bats and bushmeat, and in the parts of Africa where they're eaten, they're really popular. And in fact, this is widely consumed, and we've characterised a commodity chain that's associated with the trade of more than 100,000 of these just in one city alone. Now, we know that you can find virus signatures in bushmeat, in a Central African bushmeat market with this species. Nipevirus sequences have been detected in them. However, these are usually traded after heavy smoking as a way of preserving the meat in the tropics, and that's likely to inactivate them. So probably it's the practice of trapping and hunting them that's far more dangerous than the consumption itself. And the trade itself is quite interesting. I mean, I'm not going to dwell on this, but hunters sell to many different routes if they're commercial hunters. But there are also many individual people in rural areas that will hunt them just for themselves and their family rather than hunting them in huge numbers. But there are whole commodity chains. But because they're so popular, they're usually sold by almost soon after dawn in bushmeat markets. And so there are lazy European often, but Northern Hemisphere students of bushmeat that go to study bushmeat markets at 9 o'clock in the morning, which I think is quite early, never find bats there because they've all been sold because they're so popular. So this is certainly in West Africa is rather understudied and not well understood compared to the better way that we understand other forms of bushmeat trade. I think some of the most insightful work that we've done has come from a simple study of health-seeking behaviours in a remote part of central Ghana. So Freya Jeff conducted an ethnographic study, lived in the community over a 5-month period. It's an area where malaria is hyperendemic. She looked in both village living and also town living with some families, communities that she got to know very well over that time period. And she looked at what happened, what people did and what they described they did when they became ill. There were unsurprising differences, particularly related to income, but also to signs of disease that people found in themselves and how ill they became that impacted on the healthcare usage. But one of the things that was common between both the towns and the much more rural centres was that when people did present themselves to westernised healthcare clinics, they were usually, with a temperature, they were really just treated for malaria. Now, in many cases, the temperature will be malaria. But what was really insightful was an understanding when they came back, when the temperature hadn't disappeared. Notes weren't referred to. Another diagnosis of malaria was made, and then people went away again. And typically then, if they became ill again, they wouldn't go back. And often, if they died, and certainly one of the patients that Freya was part of the community that she was studying with died with a febrile illness, and there was no investigation made, but there was another diagnosis made. And in these really resource-limited settings, actually there is very, very little usage of any diagnostic test whatsoever for individual patients. And that means that there is more or less no detection, ability to detect emerging infectious diseases in the parts of the world, certainly this part of the world, where it's one of the highest risk areas for the disease emergence. So you don't expect a disease to be detected unless there's widespread transmission in people, which is going to trigger national or international investigation. Now, we know from basic theory that if you expect a disease emergence from animals to people in rural areas, the vast majority of cases, even for an infection like Ebola that could transmit between people, the vast majority of cases won't transmit. And so that means almost certainly that we are massively underdiagnosing every emerging infectious disease in sub-Saharan Africa, including Ebola. To supplement all of these different studies, and part of the range of studies, we've been looking at and conducting experimental studies in this large cage that is part now of Accra's small zoo, although it's in a separated part from the part that's open to the public. What we've found is this bat population is infected, even though there are only small numbers of them, persistently with Nipeviruses, which has given us real insight into how these viruses persist, because you thought before that you needed populations of hundreds of thousands of them for the virus to persist, and what this work has very clearly demonstrated is that's not the case. We thought that the cage was to contain the bats, but in fact they far prefer running around on the cage than they do to the extensive efforts that we've made to enrich the cage with wood and trees, which is a more natural habitat for them, but they adapt and they fly around this cage quite happily. We've demonstrated some primary things within this cage population, and some of them in relation to maternal immunity have been unsurprising but important observations to make. Others, like the impact of pregnancy on immunity levels for some of these Nipeviruses, have been really very, very strange, where we've seen marked increases in females but not in males at the time of late pregnancy. And we don't understand, no one understands the mechanism for that, but it provides a clear question for us to try and understand the mechanism of. So I'm just going to finish off with a couple of slides thinking about Ebola virus and some of the ways that we're taking this broader work forward, and other groups are too, because as well as looking back over 50 years, I think it's important that we think about what the future holds for us. We don't understand at all how Ebola virus persists in bat populations. We know that some transmission occurs between bat species, but we don't understand how bats may transmit it both between individuals or between species. In contrast to many other species, the virus has only been detected, genomic signatures of the virus have only been detected in bats, which almost certainly reflects the really low viral levels in infected bats, which I think a hypothesis is the case because their immune systems control the virus so much better than other host species, be they humans or non-human primates, which don't have immune systems that have co-adapted with it, co-evolved with the virus to control it, so it doesn't just kill all of those species that it's trying to survive in. So that's become, unsurprisingly, a more important area for us to focus on in the future. So what are we and other groups going to do, though, thinking more generally about the way that viruses and viruses persist in bats and so on? So it's clear there will be far greater characterization of infections of animal viruses, because genomically we can do that much more than we ever could in the past. We can do it simply and cheaply. And similarly, there's going to be greater characterization of what infects humans globally, be they in rural West Africa or urban East Africa or rural or urban parts of the developed world. But one of the key things that I predict is that there's going to be a continued realization that virus characterization sequencing is not going to be enough to predict disease risk. And I think we need to focus far more on risk factors, trying to understand what the factors promote spillover of infection from animals to humans, but also thinking about not just what promotes the spillover itself, but also promotes the ability for us to detect it. And I think at the moment the major risk factor for detection of outbreaks of disease in humans is transmission between humans, which is very, very different from the spillover itself. I think there needs to be a far greater focus on policy and responses to these spillover events, rather than thinking just about trying to understand what the virus is and what it does in a laboratory mouse or in a test tube. And thinking about the way that human societies respond to these, the diagnosis of these spillover events is a really important feature of this, because so much of the damage in all parts of the world that spillover can cause is actually the unintended consequences of the fear of these spillovers and so on. We've been working very closely with social scientists and conducting our studies of our natural science studies of the way that viruses persist in bats and how they may spread to people through spillover, in the context of public health, in the context of the ways that human and bats interact, and the way that bats interact with the ecological system that they live in. But also there's a far broader political and societal context for this. And this conceptual framework that I describe here, that we published a few years back, I think is a really important way for us to conceive and think about the way that societies respond to and deal with spillovers and their detection. I think that we're only scratching the surface in terms of our understanding of both the natural history of these infections and how they persist in bats, but also the way that our societies respond to the growing knowledge of the spillover events. And I think that's understanding that and how we should respond to these things in a way that's reasonable is a very important thing for us to do far more of. But what are the real public health impacts of emerging infectious diseases? What is their significance for public health? I think it's very important to distinguish the epidemic potential versus the endemic burden of disease. And who are these things important to? I think that in many cases, the West, the Northern Hemisphere, is far more concerned about emerging infectious diseases than public health workers in the developing world are, who are dealing with the burden of thousands of people dying from tuberculosis or HIV infection, when some of us are only worried about the possibility that a few people might die from Nipevirus in Africa, but it's never caused a detectable cluster of mortality events. I think one of the things that we need to understand far more is what the role of livelihoods is in driving spillover. It's more than just contact with the reservoir of intermediate hosts, but it's clear that its particular livelihoods are associated with far greater contact with reservoir, which puts people in some livelihoods at far greater risk of spillover. I think one of the greatest risks of spillover is poverty. And I think it may well be that for all the public health work that we want to do, trying to alleviate and ameliorate the impact of poverty across the world, but particularly in sub-Saharan Africa, probably could do far more to reduce the impact of spillover than any specific intervention that we can possibly devise. I think it's important for us to consider how our research findings can change risk and be used to change risk, but I think we have to be realistic about what some of the bigger drivers of spillover itself are. That's all I wanted to say, but before I wrap up, I just want to thank the fantastic range of collaborators, some of whom I named at the beginning, who come together for various meetings, but then between meetings we're busily getting on with our work in studying these things. This is a picture from a meeting at the Institute of Development Studies we had a couple of years ago at the kick-off of one of our bigger consortia looking at the risks of zoonoses. One of the most exciting things that's come from this work that I've been involved in Cambridge is to get involved in the capacity building efforts of the university in a major Cambridge Africa programme which links Cambridge University with other universities and research institutes in sub-Saharan Africa. I refer to a website there on the slide, but this for me has been one of the greatest ways that we can try and contribute to the development of helping brilliant people in very poorly resourced universities to do more for themselves. But it's been a great joy for me to be involved in that particular very successful programme. I just wanted to finish with a picture of a beautiful sunset in a beautiful part of the world, showing some wonderful fruit bats flying off into the distance. These bats play a really important role in biodiversity and the ecosystems that they live in. And I think it's really important to consider them as co-inhabitants of this world that we live in, rather than just the source of all things that are nasty. Thank you very much.